In this brief video tutorial, we're going to be working with the Progenitor Cell Biology Consortium Interactive Expression Viewer. This viewer allows you to take mRNA, microRNA, or methylation data that's been processed by this consortium and provide it online for interactive navigation. You can search for pathways, significant genes, or search for individual genes and their correlations. In this case, we're going to start by looking at significant genes. Here we see the viewer with the default set of genes selected. If you now go to significant genes, select an individual criterion from the menu. Here I'm going to look for endoderm versus ectoderm. These are all of the genes that are now differentially expressed in endoderm versus ectoderm versus the standard differential comparison analyses. We're now going to go to the bottom of this menu and select Enrichment Analysis and Top Gene. This loads all the genes. In this case, there were 747 genes with identified symbols. We're going to go down to the bottom here. We could do an analysis on all categories. In this case, we're just going to do microRNAs. We're going to keep the statistical threshold at false discovery rate less than 0.05. We get a large number of hits. We actually get 3,200 hits in total. But we're just going to focus on one of these top terms. So we're going to select one of these, HSA Mir 124. And we're going to look at it in more detail. If we go back to the PCBC browser, we go to the top and we select MicroRNAs. We can now download all the microarray expression profiles. We can now select for our microRNA of interest. Copy just the Mir 124 entries. There are a few because there are different versions of these microRNAs, 1, 2, 3, 4, 5' and 3' versions. And these are organized, these samples are organized typically from embryoid body through the different differentiated lineages which are available within this compendium that we'll see a little bit more clearly in a moment. So here are all the profiles for each of those microRNAs. They actually tend to be similar, which is not surprising since this is a one microRNA family and they are likely regulated in common. And we can look at these profiles. In general, we see there are a large number of peaks here and here. The peaks that are occurring here appear to be ectoderm based on these labels. And the peaks here seem to be pluripotent stem cells, which don't have an endoderm, ectoderm, EB or mesoderm label. However, you'll notice that only about half of the pluripotent stem cells have this induced ectodermal signature. Whereas, at least if you look at the green line here, most of the ectodermal samples have this signature. Since all the microRNA data is not perfect, we can go based off the assumption that Mir 124 is actually doing something important in the ectoderm. And this actually fits our original hypothesis, since we looked at genes that were induced in endoderm versus ectoderm. So the targets that we are identifying are effectively shut off in ectoderm when these genes are turned on in endoderm. Since this microRNA 124 is induced in ectoderm, we might hypothesize that Mir 124 is actually suppressing endoderm in the ectoderm. If we now look in the literature, we can find papers that now support this. I've already done a search in advance, and I can see here that Mir 124, along with two transcription factors, BRN2, MYT1L, actually are sufficient to reprogram fibroblast to functional neurons, or ectoderm.